Hi, it's Scientifically Speaking here, and today we are gonna dive deeper into blood groups or blood type, if you wish, and yeah, that's pretty much it. So let's get right into it. What's important is that we need to know the blood type in order to give a blood transfusion, in order to do transplantations, and blood transfusions are pretty common in our healthcare. That's why we need to be sure that we are like giving the person the right type of blood, and also there are several blood type systems or blood group systems. So the RH, for example, also plays a role in gravidity, in motherhood. So we will talk all about that later. So let's start with some basic information. Here we've got this important table where there are blood groups presented of like the ABO system. So this is the ABO system. And yes, so our first group is blood type A. So here we've got the red blood cells. That's what the RBC stands for. And each of the red blood cell has some of the antigen on its surface. It's a glycoprotein, and it's coded in our genetic information. So it's just like not randomly put on there, but we have it like coded from our mom and dad. So type A contains antigen that is antigen A. We will mark that with green color. So let's see. This is our antigen A. So all of the red blood cells of a blood type A person has these green antigen A glycoproteins on the surface of their red blood cells. Then let's move on to the red blood cell type B. They all will have the, let's just say, pink antigen, and that's antigen B. What about AB? AB will have both of these. And B. And what about, well, let's just finish this first. So AB will have these two. What about blood type O, you might ask? Well, it just has this, let's mark it as the black stick, but nothing on it. Now, we have covered the antigens. And also, if you have like a certain antigen on your red blood cell, you have to have antibodies circulating in your blood as well. So you also have some antibodies. These antibodies are crucial when we are talking about blood transfusions, etc. Why? Well, because, for example, blood group A has specific antibodies, and they are called anti-B. Why anti-B? Because if you give a blood group B and anti-B antibodies together, these will coagulate. To be particular, these will connect to these because they've got high affinity towards them, towards the antigen of red blood cells, the anti-B antibodies. So they just want to connect. They just want to be together because they are compatible. Or actually not compatible when we are talking about blood transfusions, but as I said, they have like really high affinity towards these. And if they coagulate, it's bad for us. Like we cannot have this during the transfusion. It's a result of like incompatible transfusions. So that's why it's an issue, because the blood coagulates, and then it cannot like transfer the gases correctly and optimally, and it's a huge issue because there's also hemolysis occurring, etc. So we don't really want that. That's why it's important to know these things. Also, let's move on to the blood type B. If we have blood type B, you have anti-A antibodies circulating in your blood. If you've got AB, you cannot have either of them because, as I have said earlier, they would coagulate. These with the A type and these with the B type. So we don't want that because then it would be real bad. What about blood group 0? Well, they do have anti-A and anti-B antibodies as well. So they have both because they have basically like nothing to coagulate with here because it doesn't contain the glycoprotein of neither A or neither B blood group. So that's why blood type A can have anti-A and anti-B at the same time. Alright, let's move on to the other problem because, as I said before, the type O has just this like stick and nothing on it. And this stick is coded by gene H. And this stick is produced just if you have capital H, lower h, or two capital H from your parents. So these two alleles from your parents. But if you have just small 2h, it's not coded. The stick is not present. So even though you can genetically have a combination for maybe blood type B, you cannot, it cannot express itself because you are missing the stick, and therefore the signature blood type A cannot connect, or B, or whatever. It doesn't really matter. So that's why it's an issue, and this phenotype is called the Bombay phenotype because if it was first discovered in Bombay. So Bombay phenotype. So imagine it can be an issue, especially if you are having some parental concerns, about fatherhood. So that can be an issue because then your partner can be distrustful and he doesn't have to trust the woman he's with that she didn't cheat because their baby doesn't have the blood group that it's possible from having these parents with certain blood group. So how does it work? We will take a closer look because it's actually interesting. Because if you have blood group A, then you are always inheriting some genetic predispositions from your parents. So if you have blood type A, you can have A from your dad and A from your mom, or you can have A from one parent and 0 from other parent. So this A allele is basically stronger than the O allele, so it kind of overpowers it. So that's about it. If you have a blood type B, it's the same principle. You can have either B from each parent, or you can have B and O. If you have a blood group AB, the genetics behind it is that the A and B allele are codominant. That means that if you have A and B, you can only have A from one parent and B from other parent. If you've got O, you can only have O from one parent and O from other parent. So that's why the Bombay phenotype can be an issue while we are talking about fatherhood and the identification of a father based on blood groups. Right. Another thing is the Rh factor that I have mentioned before, because the Rh is mainly coded by gene that produces some D antigen. And if you have capital D and lower D, and if you've got two capital D's, you get the Rh plus blood group. But also these are another structures on the surface of red blood cells, like the products that are coded by these genes. So the Rh blood system is also concerning some surface structures on the red blood cells. But if you've got two lowercase d's, then you are Rh minus. So as you can probably imagine, the Rh minus is there's like lower probability that you're gonna have this type of this Rh blood group, and that's gonna be an issue because if you got pregnant mother, let's imagine that this is like a pregnant belly, and she's got the uterus in here, and then a baby in here. Oh wait, it should be head down probably. Right, and you've got the baby in here, and let's just say that the father of the kid, this is the mother, and the father is Rh plus, and the mother is Rh minus. Let's just say that he's got one capital D, one lower D, and she's got two lower D's. And this father gives the capital D allele to his child. So the child is D and small d because there's just only one option that it can get from his mother. He can only get, or the child can only get one lowercase d from his mother. So then the kid is Rh plus. Well, and it's an issue because or usually not for the first pregnancy, but mostly for the second one and all the other ones, because when there's like childbirth occurring, there's like a little bit of blood mixing of the babies and of the mother's blood, because during the pregnancy it's separated because of placenta. So it's not a huge issue if there's not any like specific problem, some complication of the pregnancy. But this can be an issue after the first birth, because then the mother's and the children's blood or child's blood comes into contact, and the mother is starting to produce anti-D antibodies. So that's why it can basically, these antibodies, they are preserved in the blood of the mother, and if there is another pregnancy, then these antibodies can attack the erythrocyte red blood cells of the second child, causing them to hemolyze, and then the child can die because of hypoxia and just like not enough oxygen present in his body. So that's why it's such a huge issue. Now, to sum it up, let's take a look at some practical things, because every time you want to give a transfusion to a patient, you need to do this bedside test with each and every one transfusion. Bedside test. It's called bedside test because it's actually done by bed of the patient, and it's really quick, and you just always need to be safe and confirm that the blood you're gonna give the patient is actually compatible with his blood. So here's the test. You've got some anti-A and anti-B. Here you put the anti-A antibodies. It's like the blue fluid, and here you put the anti-B antibodies. It's the yellow fluid, and into that you give the blood of the this is the receiver who is receiving the patient, and this is the blood pack that you're about to give the patient. So yeah, let's say we've got, this is the receiver and this is the blood pack, and now we've got a situation because how do we recognize the blood group from these two need to be compatible. They need to be compatible with each other so we can give the receiver the right blood. And if we put our blood and it coagulates, which you can see here, it did coagulate with anti-A, it means that antigen A is present in the blood. Here it's starting to coagulate with B as well, which means that this is AB blood type, the first one. What about the second one? Well, the second one, it's actually you can see that with A it's coagulating, so it has A antigen for sure, and with B it's not, so it does not have the B antigen. So this is blood type A. Now, can an AB recipient receive A blood? Can he receive A blood? That's the question. Let's take a look. Well, we've got the AB recipient. And it's pretty much universal recipient because, as we have mentioned before, it does not contain any antibodies in his blood because they would attack his own red blood cells. So, therefore, he can accept blood type A. So that's about it from today's video. Hope you enjoyed it. I will be happy if you give me like and subscribe. And see you next time.